discovery dialogue session held at the Research Institute at the Hospital for Sick Children in Toronto. My name is Steve Scherer, and I'm the Chief of Research at SickKids. And today's guest is Professor Lap-Chee Tsui. Lap-Chee is well known to many of us as the scientist who led the team after a decade-long search that discovered the gene and the protein that is involved in cystic fibrosis. Lap-Chee has remained a distinguished senior scientist at SickKids since 2002, when he became the 14th Vice Chancellor, which is the equivalent of being President of the University of Hong Kong, a position he held until 2014. In addition to his SickKids appointment, Dr. Tsui was founding President of the Hong Kong Academy of Sciences and held that position from 2015 to 2022, and he holds many, many other active appointments around the world. So in today's session, I would like to focus our discussion on those very few special months over the summer of 1989, 34 years ago. I hope your memory is still good, Lap-Chee. When the three papers that were concurrently published in Science on September 8th were being written. So the title of the session will be recorded as Prose Under Pressure. There's much written in the literature about the details and milestones of the decade-long search for the cystic fibrosis gene, and there's a wonderful book that was just published recently called Breath from Salt. If you're interested, you should read this book. It talks about the gene discovery going right to the development of therapeutics. But very little has captured what was happening behind the scenes in writing the actual scientific papers. For most of us, it would be unimaginable to think that our research would lead to back-to-back cover issue papers published in the world's leading scientific journal at the time, which was Science magazine. Of course, these papers collectively are the most highly cited ones in all of the SickKids history of research. Congratulations. And the discoveries of the gene and the protein they describe has led to new diagnostics as well as novel prescription medicines to help hundreds of thousands of people afflicted with cystic fibrosis worldwide. So before I go to the questions for Lap-Chee, I wanted to give a brief chronology of events leading into 1989, in particular for those of you who may not be familiar with the story. And I know some of you in the audience weren't even born at the time, so so just bear with me a little bit, and then we'll get to our guest speaker. So in 1980, Lap-Chee came to Toronto from Oak Ridge Laboratory to do, I think it was a postdoctoral fellowship, and he can talk about this later, at SickKids, working with Manuel Buchwald and also working closely with Jack Reardon. In 1981, I think you were given your first laboratory bench and started setting up molecular biology technologies here at SickKids, culminating in the idea to adopt a genetic linkage mapping approach to find the gene, because there were no biochemical clues available at the time and no chromosome abnormalities available to kind of guide the search along the DNA, similar to what was happening in muscular dystrophy work here at SickKids. In 1983, there was a very famous paper using linkage mapping approaches published showing the first linked markers in Huntington's disease on chromosome 4, providing some guidance on how you might do the same linkage mapping for cystic fibrosis. And ultimately, Lap-Chee performed many experiments with technicians, some of which are in the audience here today, and ultimately found linkage with DNA markers to the cystic fibrosis locus, and just shortly thereafter mapped those linked markers to human chromosome 7. I think your favorite number is, your lucky number is number 7. We can talk about that later. And these papers were published in 1985, and the first one was in Science, if I recall properly, but there was also another paper in Nature, and I think we'll come back to this as a discussion point a little bit later. 1986, Joanna Romans, who's also a senior scientist here, emeritus now, arrived at SickKids. 1987, Batsheva Karam arrived, and we featured Batsheva and her husband A10 Karam in a Discovery Dialogues last summer when they visited Toronto. You also initiated the collaboration with Francis Collins in 1987 and conducted what I would call a saturation mapping approach to identify new genetic markers. I'm getting to the end in a second. In 1987, something very eventful happened. There was a paper published out of Bob Williamson's group at St. Mary's in London, England, describing the first candidate gene. It was published in Nature, and many people thought that was the cystic fibrosis gene. So I want to come back to that a little bit later. And then ultimately, I started as a graduate student in 1987, and that's relevant. I wasn't working directly on the cystic fibrosis project, but I was developing new technologies like yeast artificial chromosome mapping that would hopefully speed up the way, but they found the gene first. But I actually had a very unique vantage point to make observations of what was happening behind the scenes. And some of those have led to the questions that I've been waiting for 34 years to ask him in front of this audience. So 1989, early in that year, a gene fragment was identified, I think, by Joanna Romans. It was conserved. It turned out to be part of the cystic fibrosis gene, but nobody knew at the time. May 9th, I think it was at 6 p.m. I read in this book here. I'm going to ask you if you remember the day of the week later. The first 3 base pair deletion was identified, which would later be called Delta 508. And August 7th, according to the 3 papers that were ultimately published in Science, which are shown here, you submitted, so again, August 7th, you submitted 3 scientific papers describing the gene mapping, the identification, the cDNA and protein, and ultimately the genetics to prove you had the right gene. Now, this is amazing. I hope, for those of you who are publishing, the papers were submitted according to this issue in August 7th, and they were accepted on August 17th, the first paper, and the second 2 on August 18th. So we want to hear a little bit about how the heck that actually happened. So again, I hope we can focus our discussions on the details of what was happening behind the scenes through the summer of 1989, and particularly with respect to writing the papers. My hope is that some of the lessons that you share will actually be relevant to the next major discovery here at SickKids, and how you work the journals, how you conceptualize writing a paper under pressure. There's time pressure here. And ultimately, I think there's some key lessons and kind of facts to be shared that we don't want to get lost through the history of time. So we're really hoping you'll share with us what was going on in your mind at that time. Okay, so on to my questions. In the news and views leading into the description of the 3 papers that you wrote, there's a great quote at the very first paragraph. I don't know if you remember this or not, but it was from, and this is written by Gene Marks, but the quote is from Luke Kunkel, who's at Boston Children's, Harvard, who I think was probably a reviewer of one of the papers. And he says, the data are virtually irrefutable that they have the right gene. So here's my question. When did you know you had the right gene? And when did you start to conceptualize the writing of the papers? And how did that play out?
Thank you, Steve. It's really nice to be back at SickKids. And even though I never really worked in this building, it's so nice. And I worked, as Steve started earlier, on the 11th floor of the Elm Street wing, on the bench that well nicely provided for me to start this genetic mapping work. And then later on, the gene finding was actually done on the 9th floor, right? Also on the 11th floor, I should say, 11th floor, with all the other researchers around, and then to keep that a secret was not easy, right? But people knew that something was going on. So when did we first realize we had the right gene? No, it was not immediately. In fact, Joanna would have told you, even 6 months after we submitted the paper, we still worried that we did not get the right thing.
Right.
Okay, it was very scary because we had a 3 base pair deletion, even though we came up with the argument, so-called evidence. I come back to that. It's an interesting story behind that too. We had no idea we had the right gene until the next mutation was discovered. It was so stupid at that time. Why didn't we look for a second mutation? We kept trying to prove that the 3 base pair deletion was a cystic fibrosis mutation. Mm-hmm. I mean, come to think of it, so lesson number 1 is that don't believe in yourself so much. And then in terms of approach, let other people work on it. If we had leaked the information to Gary Cutting mm-hmm. ahead of time, he could have found the mutation. He's from Johns Hopkins.
Yeah.
He did not have the whole gene information. He just used the cDNA information and amplified a segment, an intron of it, and found second mutation. When we saw the mutation, we said, how come we did not look for it ourselves? And spending all this time worrying that we did not get the right thing. Okay? And how do we convince the science reviewers? Okay, of course, we collaborated with a lot of people. And then sometimes I say, genetics cannot fool people, and this time we actually fool people. Okay, when you get a odds score or the probability score of 10 to the minus 57,
Right.
what do you say? Oh, got to be right, 10 to the minus 57. And then I did not ask Aravinda Chakravarti how he came up with the number. He said, I just cut it from there and put it down, and nobody argued. Because he was an expert.
So Aravinda Chakravarti, who was really a junior statistical geneticist at the time, was a friend of Lap-Chee's from their PhD.
Pittsburgh. Yeah.
And when did he come up here? That's one of my later questions, but let's jump ahead. When did he come up here to kind of look at that? And I'll just say, just to lead into this, there's a great quote. It builds from what Lap-Chee said. Aravinda Chakravarti said, there's a 1 in 10 to the 57th chance of it not being the mutation to cause cystic fibrosis.
Yeah.
It's an interesting way to kind of phrase that.
Right. So it is a really his reputation in the field, and he's known to be his mathematical skills. But later on, he failed on me. But then it can come back to that. So nobody argued, but in fact, there was no calculation whatsoever.
Yeah.
He just came up with a score.
Okay, so back to the question at hand. See, we have a special relationship. Were you convinced? Because in the book, in my memory, is you were the most critical of the data. And were you convinced it's time to write the paper?
Okay, so when you had any hint of a step, you start to think about. So if somebody else publishes a paper, what do you do? So you scrap whatever you have, read ahead of time. So constantly in your mind, you have the data well organized and your arguments well made in your head. So writing the papers were actually very easy. It was very easy, especially for the first one. It was very straightforward. I just described the steps and so on, and then very descriptive. And then the second paper was a bit more difficult because before you had the gene in hand, before Jack actually did the analysis of the predicted protein, it's very difficult to say anything about that, right? So, but I still had a lot of time because the first part of the gene we cloned was actually exon 1. Okay, when you think about cloning a gene, you have a cDNA approach, you come from the 3' end, that's the back of the gene, and you move forward, right? So this time we had the front of the gene, and then somehow we could not get the back of the gene because the gene itself, the cDNA, was not very stable.
Right.
Okay, so it took us a long time to finish the rest of the gene and make sure that we had the whole gene to publish, right? And then there's no other gene hidden or somewhere else. So we had a long time. So I even had time to draw a model of CFTR, the protein it predicted. Okay, so we had lots of time to prepare the second paper. The third paper, it came about because we got into some argument about authorship. Okay, I'm sure that you're going to get into that.
No.
Okay. No. So how do you come up with the third paper? In fact, the third paper, I sometimes say it's the most important paper because it's genetics. Okay. So and then the proof was in the 10 to the minus 57th, right? But a lot of things in there, a lot of things we could project to the future from the genetics. Anyway, so the third one came about very quickly, but since we knew the data day in day out, and we had a lot of time from the first identification of the 5' end of the gene, you mentioned sometime in May, and we submitted a paper in September, so a few months to get the rest of the paper done. So plenty of time.
So in the book here, they talk about when you picked Aravinda, flew up from Pittsburgh, and you picked him up at the airport, and I remember him coming to the laboratory, and then he wanted to leave, and you wouldn't let him leave. And how fast did you write the third paper? Because
the third really paper was already written before Aravinda was here.
Okay, I have to remind him of that when I see him next.
Of course, he knew that too.
Yeah.
The reason is that I wanted him to see the data firsthand. I could not ship the data or the charts to him. So he had to come up to see the data himself.
Okay.
Okay. So to package the rest of the paper, I had to go through him.
Yeah.
To make sure that they all make sense. In fact, the third paper was cut short by a hypothesis at the end. The hypothesis was to use the haplotype mapping to clone a common disease gene.
Right.
It became very popular for small period of time, as you say. Okay, and I asked Aravinda to come up with a mathematical solution to that problem. Because you can see it by eyeball, you can borrow that Science magazine, the cover. Mm-hmm. Okay, if the audience can see the magazine, that these are the haplotype, the diagrams about haplotypes. You can see how the haplotype shape up and narrow down into a smaller region. That's how by eyeball I say, oh, the gene must be there, right? But then you cannot. That's easy afterwards because you know the gene was there. You know the. Right, but how do you know ahead of time? You must have some mathematical sort of calculation and so on. And he's supposed to come up with a mathematical solution to that. And that part he wasn't ready, and he did not really write up the paper properly either later on. And then another group, the Eric Landers group from Whitehead, actually came up to cloning his first gene based on haplotype mapping. So that part of the discussion part was cut out because the editor said, hey, this is your discussion, so it's not about this paper, but I thought it's damn important. Right. So you won't let me put that part into the paper. Then I thought about a great idea, put it on the cover. So for those of you who remember the Human Genome Project, the next stage was actually called Haplotype Mapping Project, and the first reference, I think, or first three references, is the third paper of CF that lays out how you develop a linkage disequilibrium or haplotype map of the genome, actually, and it's cited as that. But so they chopped that part out of your paper. Really interesting. One of my later questions, but I'm going to jump to it right now, is in the first paper. You had a section, the first paper is the chromosome walking and jumping to find different genes and sort out the right gene. You had a section in the paper that was, let's see, what was it called here? I have to find it. Let me look in the notes here, but sorry about that, Steve. Yeah, you threw me off. That's okay, Lap-Chee. Lessons from the search. Do you remember writing that? This isn't the first paper. Do you remember that part? No. It's kind of in the discussion. Okay, I'll remind you. It's kind of what you just said about the third paper. I'm sure you and Francis both wrote this because it lays out in there how you need families to participate, you need collaboration, you need multidisciplinary approaches for big projects, and how for most human disease genes, there will not be a chromosome abnormality to kind of mark the way forward, and you need to use this approach. And in fact, it doesn't say it, but it lays out the plan for a Human Genome Project, actually. Okay. So there you go. That was brilliant. And in fact, for those of you who don't know, in U.S. Congress, James Watson and later Francis Collins, who became the head of the Human Genome Project, they argued, based on the cystic fibrosis story that there should be a genome project and a sequence for everyone so you could find all the different 6,000 disease genes. So that's laid out in paper number 1. All right, let's keep going. I want to get back into your head here. From my observation, it was a once in a lifetime experience because you were, I use the phrase living in the moment. Everything, you were so focused. I remember you'd be at work 8 a.m. till pretty much 10 p.m. every day, and after 6, you're sketching out the figure 7 in paper number 2, which is the protein structure. So how did you kind of stay focused through that time with everything else going on? There were still experiments going on. You were starting to write papers. How did you kind of keep everything on track? Well, I did not work in a lab myself, so that's why I had all the time. How do I put it? I did not start at 8 o'clock, just to correct you. Probably 10 or something to avoid the traffic. The reason is that we did not stop at 10 o'clock either. Okay. And Joanna Robbins, you just mentioned. Yeah. And then a number of others, they live somewhere outside the city because the rental was already expensive then. So I had to drive them home because there are no subways. Okay, so after I got home, usually it's 2 in the morning.
Yeah.
So it was difficult for me to get up to get back to work at 8. Anyway, so we did maintain that pace for quite some time. So since I did not work in the lab, so I have all the time to think. And then from time to time, I get Joanna and Bacheva, and then at that time also Richard Rothmahal and others. And then you probably seen all the commotion in my office, a very tiny office. So we had in there and had conversations, right? To think about the next moves, especially after the report of this Nature paper in 1987, where the Bob Williamson's group reported that they had a candidate gene. In fact, it was a very embarrassing moment. I think some of you might have read some of the things I wrote or talked about in other times. I was attending a Royal Society conference in London, and on that day, he published the Nature paper. He gave me a preprint to read that he had the gene, and he asked the organizer to let him have a podium to talk about his gene, whereas I was invited to describe how I was going to clone the gene, right? So very embarrassing moment. But I quickly looked at the paper. There was no gene information there. So he might have the gene in hand. He might talk about it in his talk. So it was a very difficult decision when the organizer asked me whether I would give him a slot. I said, sure, he could have the time after I speak, and I'd like to hear what he had to say too. But he did not present any information about the gene. And then, in fact, in the published paper, there's no information about gene. But we had to deal with the fact that he has something exciting. So a lot of groups in the world actually stopped chasing after the gene because Bob made the announcement that he had the gene. So the most difficult time was actually to speak to Joanna and Bacheva, to some extent Jack Reardon as well, and then Francis Collins. You mentioned that we had to continue.
Right.
And then the reason is that we hadn't seen the gene yet.
Right.
Okay. Even if he had the gene or the Bob's group had the gene, that we still have things to work on. So I have also to convince Joanna and Bacheva there are lots of work in front of us because we knew so much about the genetics of the disease, and then we have so many things to contribute. So don't give up. Let's move on. And if we see the paper, then we believe they had a gene. But fortunately, we moved on, and the other groups all dropped out. Okay, and Noah here, who would remember that most groups dropped out.
Yeah.
Right, and then so we were the only group working on the CF gene. Even Bob's group fell apart because all the postdocs and students got jobs. They left the lab.
Yeah. Yeah. So I mean, they essentially published the equivalent of paper 1, and then, as you said, when you got the right gene, you published paper 2 and 3 to prove you had the right one. Yeah, irrefutably proved.
the number 2, the second paper, that the mainly about the cDNA, the sequence, what it presents, and then the where the mutation, the location, deletion of a single amino acid residue, and all that was described in the second paper. And then so it's just a sequence. Yeah. So sorry to interrupt. So just to come back, so I was going to ask you about the Royal Society experience. In my notes here, it must have really got you charged up and got the fire in your belly that you knew it wasn't the right one, but you had to prove it and keep moving forward, as you said. What was the other lesson from the 1985 linkage papers? Because you kind of got burned there a little bit, although you got the paper out first. And my question is I think the linkage paper was in Science, right? Yep. Did that kind of motivate you to publish the cloning paper in 1989 in Science also because it was a good experience with the journal, or what's the story? Or tell people the story. The members in the audience, I think Science and Nature, they were in competition all the time to get the best paper in those days, okay? And now, of course, there are many other ways to publish. And then I remember that Nature would have a little bias towards the British scientists, and then Science, on the other hand, I had a good relationship with a senior editor then, who was already 80-some years old. He taught me how to write a scientific paper. Because I never use a single person, singular. Okay, don't
First
say
person.
I in the paper. And then don't say you are the first. Okay,
do you remember his name?
History would tell.
Who is that?
That's,
Okay.
no, there's Eleanor Butts.
Yeah, okay, right.
Eleanor Butts, and there was an interesting episode afterwards, but then
We'll come back to that.
Okay, so she taught me a lot of ways to write a scientific paper. So young man, I see that you're a good scientist, but you have to improve your writing. And so I learned how to write a proper scientific paper, very objective, and then no extra predictions so on, although we try to squeeze something in there later. But so she really said, what's your message to deliver, right? You have to make it very clear. And 80-some-year-old and still working very sort of hard in the office.
Yeah. Okay, so go 1985. So just in a minute, just kind of set the stage of the linkage paper. So you're collaborating with Collaborative Research, which is a company in the States. They had markers. They provided some. Noah and some of the technicians in the audience were doing the mapping and what happened then? It comes into when do you pull the trigger to publish?
No. The thing was it was very interesting. Just to keep the record straight, and then the three technicians who were working on the project did not include Noah at that time.
Right, it's true.
She worked with Jack on the cDNA side of things.
Yes.
And then later on joined the genetic markers and so on. But anyway, and chromosome walking even. But anyway, what was the question? When
did you're going to publish the paper and then the mapping?
So Collaborative Research advertised for the fact that they had lots of genetic markers. They're going to develop all the genetic markers throughout the genome so they would be able to map any genetic disease genes in the genome or in the human for human diseases. So where we had the families, I think we had enough families, by the way, using small families to do genetic mapping. It was also a surprise to many geneticists. You mentioned about Jim Cosella's Huntington family. They're huge families. Yeah. People knew how to do odds calculations with large families. But nobody was interested in small families, that CF families with 2 affected individuals or 1 affected and unaffected, that type of small families. And but it worked, okay? Yeah. So it became what do you call it, unexpected finding for the human genetics field even.
Right.
But nobody actually was using software to provide loss scores for such small linkage families. I had to do everything from scratch, from first principle. I make some wrong assumptions here and there, so the numbers were not quite right. Okay, but the company had the DNA markers, and we had the families, and then we had to put them together. So we thought the collaboration would be good. But companies, they had the objectives. They want to clone the gene as well. So once the first batch of DNA markers was shipped to us, we're supposed to send all our DNA family materials to them as well, so we can both proceed at the same time. But we're way ahead of time. We had all the gels, southern blot. So in fact, it's not southern blots. They call DBM paper in those days. But anyway, so it's a blot that can be reused.
Yeah.
Okay. So it's so easy for us to put the markers on a piece of southern blot and look at the results. Okay. So the first set of 12 markers they sent, we put it on the plot right away. And then there was one out of 6 that show a positive, sort of very respectable lot score, the lot of odds, right? So 2 point something, right? So very respectable.
Yeah.
And so very excited. So we immediately notified the company to say, okay, is there any marker nearby? But they had no idea. where those markers were generated from, and they had no idea it was on chromosome 7 either.
Yeah.
So we were asked to hold the information, because if the information was leaked out, okay, anybody could jump on the chromosome 7 library and then get markers and so on. So that's not what they want to do. But as an academic laboratory, we want to get help from other people as quickly as possible. So there was a little tension.
Yeah.
Okay. So finally, it was not we who leaked the information out that the marker that they provided us was a chromosome 7.
Right.
Is one of their own advisors attending a meeting in Germany leaked out the information. So the whole world knew about the CF marker was on chromosome 7. And
Before
then
it was published. Yeah.
Bob Williamson and another group by the name of Ray White, in the Salt Lake City, they were at that human genetics conference. They heard about this news, immediately called back to the lab, pulled out everything they knew or they had for chromosome 7, the markers. Each of them found a marker right next to the CF gene.
Yeah.
In fact, the two markers were flanking the CF gene, as we learned later. And our marker shown a positive linkage score to the gene, but was really miles away, 10 centimorgans away on the genetic distance. And those two markers, with less families, they were so tightly linked, it's only 1 centimorgan away. So therefore, the two are much more useful than the chromosome 7 marker that we identified initially.
Right.
So knowing that the two other groups were submitting the papers to Nature, so we had to convince science that, look, you
got to move fast.
That you had to publish our paper first because they got the information from us.
Yeah.
Okay. So science reviewed the papers very quickly because it's very straightforward. So at a positive log score of 3 point something, they published the paper a week after the Nature papers.
Yeah.
Okay. So Nature papers came out, but Nature thought it was really unfair.
Yeah.
Nature thought it was really unfair, but they have the editors too. So they knew that the mapping was done by the Toronto and the Collaborative Research Group. They called us up, okay? And in fact, I convinced the Collaborative Research, you have to publish your mapping paper, okay? Even though the scientific data about linkage was submitted to science, they still reviewing the paper, but you publish the marker, which is located on chromosome 7.
Yeah.
As a paper separately. And they accepted it. And that's almost without review that they accepted the paper. The marker was on chromosome 7. When the science paper came out a week afterwards, in fact, the week afterwards, still a week ahead of the original published schedule.
Yeah.
So in those days, I mean, I learned how to deal with the senior directors, senior editors, right?
Yeah.
So very, very exciting.
So that was 1985, and he already
85 only, then 89.
89, yeah, but he probably the only person who published pretty much the same data in Nature and Science. Okay, so that's great. But I think, when you're going through that 1985 experience, you had to figure out when to pull the trigger, and it must have been driving you crazy through the summer of '89. What if I have the same experience with someone leaking some information out? And just to set the stage, 1989, PCR, polymerase chain reaction, was being used in the laboratory, but not ubiquitously. It came out in, was it 87, 88? But there was the threat that a probe or any sequence information could be amplified, and a competing group could move fast. And as Lap-Chee said, he was competing against some of the biggest labs in the world at the time, and you didn't mention names like David Botstein was involved early on.
That was the 85 paper.
In 85, yeah, and Eric Lander and many others. So there were a lot of groups that could move fast if something was leaked.
Yeah. There's no doubt about it. But then, they're talking about history. The first PCR machine was not in our lab.
No.
The PCR machine was Joanna Romans doing this.
Yeah.
3 water baths. Okay. And then the first PCR machine was the next door in Roy Gravel's lab. We had to borrow some time from his machine to do the first real PCR experiment with the machine.
Yeah. So to come back now, we'll move back to 1989. I wanted to maybe just ask you, just to comment very briefly on, you were kind of the conductor putting these papers together, and you had Francis, of course, input, and Jack Reardon, and Joanna, and Batsheva, and Manuel, and Ron Wharton's actually acknowledged in the last paper. He's the only person acknowledged for contributions in writing in all three papers, actually. I found that interesting. Ron was the head of the genetics department at the time. So how did did people put together different figures and tables, and you spin it all into the text together? What was the plan there? How did that work?
No, I must say that I was pretty much the person who put everything together because I had nothing to do. Finally, I beat up Joanna for the full-length cDNA, and then the rest of the lab to come up with more. So the linkage, not linkage data, the mapping, not mapping the patient data, and then that's very important for the third paper. And then in the meantime, I also communicated with a lab outside of Toronto to make sure that we had other proofs, other set of data to support us. So we communicated with an outside lab, not the Francis Collins lab, but with his, of course, agreement too.
Yeah.
Yeah. So a lot of these things, because I had a lot of time to plan, so I was able to put together the diagrams and so on. You know that I like the drawings.
Yeah.
I draw all the diagrams, pictures.
I remember.
Even paste together the gel lanes, make sure that nobody gave me a...
Do you remember I used to go to McDonald's for you to get you food to feed you late at night?
Yes, indeed.
That was my job. There used to, for those who don't know, David Malkin's in the front, he'll remember this. There used to be a McDonald's on University across the street that had a grand piano in it. Right? You remember that?
No.
Oh, just, okay. Well, I used to go every night to get you food, to feed you.
David was playing there?
No. Okay. So then to go back to the history, the papers, so let's get back to the submission dates and how quickly, and then the papers were supposed to come out on September 8th, I think it was. Then they were leaked out to the press. The media came out about 2 weeks before that. I have the exact dates here, but Dan Koshland, who was the editor of Science, then actually allowed the press release to come out 2 weeks early because I think it was Thomson Reuters or Reuters had actually started to release things. There was a phone call apparently that happened with a graduate student in Francis's lab, and the story spilled. So in fact, there was a release, and this could have happened perhaps months before, and you could have been scooped. And what were your thoughts, and what was kind of happening around that time?
No, not much you can think about. It's just to keep moving, right? Mm-hmm. So things happened, and then you could not really blame pointing fingers because it was already too late. Yeah. So we just had to convince science to let us move forward, right? And then to organize the press conference and so on and so forth. Yeah. I still remember that the sequencing, the waveforms we submitted was not accepted by science. Is that all these machine-generated sequencing results, they're waveforms, right? So even though the machine is obsolete now, it's from DuPont, and beautiful pictures, they will not accept it. Because the reviewers didn't understand what it was. No, never exactly. Seen it. They were really traditional at that time. Yeah. They have to have a gel. I still remember Cy Locker was standing in the back there. Mm-hmm. They would repeatedly run a gel to perfection. Yeah. Okay, to studying the 3 base pair deletion. So we had plenty of time to play with diagrams and figures. Not that we were so rushed to publish. And then, in fact, all the timings were good, except for Eleanor Bus, because we rushed a week ahead of time. So this old lady, I'm sorry to use the word, she actually came to supervise. Yeah. She came back to supervise the whole thing. And then the whole bunch of us actually flew a private jet from Toronto, the press conference to Washington, D.C. Right. And then when Francis, I, and Jack went to the press conference, and then the rest of the people had to sit in the library of the Science magazine to continue to do the galley proofs. And of course, everyone had to check every figure, letter, and so on. Because in those days, not the electronic typesetting, it's still by hand. So imagine. So Eleanor tripped over one piece of luggage, broke her hip.
Oh my gosh. That's the first time I've heard that story. So the first submission was really on August 7th, and then
I don't remember the exact.
But it was that fast, a couple of
Yeah.
weeks.
Yeah.
And so many of us teach these papers as classics in the courses we teach, and you have to read them. They're really beautifully written. It's hard to believe you could have actually not had to go through multiple edits and reviews. So they never got really peer-reviewed comments back. You just kind of said it's accepted.
No, because first of all, this paper has gone through quite a lot of iterations already amongst ourselves, right? So when you're waiting for the results, you always read the papers, right? So everyone was reading the papers, and Ron, he was contributing to reading of the papers too. So I had to let the department head know that we had some exciting things coming up, right?
Yeah. So take us back. So 34 years. You used to always take summer vacations. I remember you had 2 young boys and your wife. And did you take a vacation that summer? If you did, how the heck did you have any focus? I can't remember now.
As I said, I did not work in a lab.
Okay.
So,
but July and August.
Joanna, on the other hand, really did not take a vacation.
No, I know.
Yeah. So, and not vacation, not going back to New Brunswick at all. Right. But then she decided to take a time off, right, to go camping with her younger sister in the Rockies, Canadian Rockies, by the way. Okay, and then we had a hard time finding her when we had the press conference. She couldn't come back in time for the press conference, so she missed the whole thing.
Yeah.
So she took the vacation then.
So for those of you who weren't, there were a few people in the audience who were here at the time. There was a press release at SickKids in the main auditorium in the main hospital, and then right after you got on a plane to go to Washington, they had a release out of Washington, D.C. It must have been a hugely special and satisfying time for you. You spent 10 years and you fulfilled what you set out to do 10 years earlier. And so what did it feel like making that discovery? I think you were 38 at the time.
Yeah, so not quite 10 years. So it's only 7 years.
It's kind of like a hockey player getting 100 points. Will you ever do it again? What were your thoughts? And
oh, it's interesting because when I was doing graduate school, I was proposing how to clone a disease gene. And there was too much ahead of time, proposing using cDNA and all that, and I got a C from the professor. Years later, I went back to Pittsburgh. I said, Professor so-and-so, you gave me a C. Anyway, so that was actually, I can say, satisfying is one thing, but then I think realizing that you have to take a different role in science, and you have to go out to speak to a lot of people. And the 1985 story was particularly sort of worthy in my mind. That is how to speak to the public about genetic mapping. And I still remember the anchor of CBC, the lady, I think Eve Savory. I forgot his name properly.
Barbara Frum?
No.
Yeah.
CBC.
Canada.
Okay. Yeah. So she came to interview me, and then I tried to explain linkage, finding the gene, a marker, DNA marker, defining locus. How do you describe that? I had trouble, right? So she particularly said that, try to explain to your grandmother what you have found. And I said, forget it. In front of the camera, I said, it's impossible.
Yeah.
Okay, so then later on, I said, if you don't do that, you have other people talk about your own research, and then even though they have cited your work and give you all the credits, it became words from their mouth. People can easily misunderstood that work was actually done by the person who is describing your work. So I learned it the hard way. So I started to tell myself, if there's something important, get ready to explain to a lay person. Right? However difficult it is, whatever analogy you have to use, so you have to do yourself.
There's been lots published in the literature, and in the history between 85 and 87, there were many news and views published on who did what, and it all validates everything you said today. I mentioned at the beginning this book called...
It's interesting. Those validated what I say. I validated what they said.
Breath from Salt, it talks about how right after the actually part of the gene identification, you would release probes for genetic mapping and diagnostics. Peter Ray here at SickKids used them in early diagnostic testing. And of course, the predicted protein structure formed the basis for new therapeutic development. And now there's drugs that are being used here not in trials. They went through clinical trials and actually to treat our patients now. So everything that was predicted in the paper came true, which is just unbelievable. It's a trilogy. It's a great story you told. It's understandable. My last question to you is there anything you would have done differently in that summer of 1989 that, based on your experiences, that you want to share with the audience?
First, I couldn't think so far back in 1989. But if there was anything that I could have done myself as after cloning a gene, I said one thing earlier, that is look for the different mutation instead of focusing on single mutation. There was one thing, and I don't have any regrets. I should put it that way. Mm-hmm. I think people ask. You asked me the question earlier about the authorship, and then how you came up with the arrangements, such and so on and so forth. I still remember that I had to convince Francis Collins that Jack Reardon actually gave a lot to the project. Mm-hmm. He made a cDNA library from his own sweat glands. Okay, and you found the first piece of DNA from the library. Mm-hmm. And then it was Noah's colleague who came up with a 10-1. And then all these things is without the collaboration, we could have done it ourselves. And also, people, you cited the last paragraph in the first paper. In fact, I still remember that having the concept reiterated times and again in front of the general public is about the collaboration among different researchers, doctors, nurses, and patient families in a study like this sort. Without the help of the Canadian Foundation at that time, the CF Foundation, this could not have been done. I still remember that there were CF doctors who look after CF patients in this hospital did not believe in basic research, because it was so far away from what they could see. They have patients in front of them dying, so they'll look after patients. So they said, well, save the money, give it to us, we can save the patients. And for you, you just waste the money. But when they turned around and they collaborated with us, it was really really something that I treasure so much. That the basic research and then working together actually has been a very important value of having a research institute within a hospital. Mm-hmm.
Well, I think we should probably close on that important note. Thank you so much. I got most of what I wanted out of you. So I hope it's not the last time, but this has been really enlightening, and I think we've all learned a lot. And thank you for everything you've done for science and for SickKids over the years.
Okay.
Thank you very much.
Thank you, Steve. You didn't ask them if they have any questions.
Now we're off camera. I think I don't have my clock. So anybody have any questions here? Some of you are